Hello, and welcome to today's seminar. I'm Beth Mashesky, Senior Scientific Specialist at the Illinois Sustainable Technology Center and Seminar Co-Organizer. Our next seminar will be on October 25th from noon to 1 p.m. here at ISTC. It will be on air quality contaminants and their sources, presented by Vishal Verma from the University of Illinois. I'll ask our audience members here to please silence electronic devices as we are recording today's seminar. We will hold all questions until the end, at which time I will bring around the microphone so that those online can hear your questions. For those online, you can type in your questions at any time, and we'll answer those at the end as well. I have one more announcement related to today's topic. The Illinois Sustainable Technology Center will be co-hosting the 2019 Emerging Contaminants in the Environment Conference with the Illinois-Indiana Sea Grant on May 21st and 22nd. In Champaign, Illinois. It will cover topics like PFAS in today's seminar. It will also talk about pharmaceuticals and personal care products, microplastics, and many other emerging contaminants. The call for abstracts will open later this month. You can find more information on the events page on the ISTC's website, or you can email me at elm2@illinois.edu. And with that, I'm very pleased to welcome today's speaker, Matt Simcik. Matt is Associate Professor in the Division of Environmental Sciences at the University of Minnesota. He earned his PhD in Environmental Science from Rutgers University. His research at the University of Minnesota focuses on how and why organic pollutants get into the air and water, especially in the Great Lakes region. Please join me in welcoming Matt.
Can't tell colors. Did it go?
Yeah.
Okay. Okay, thank you very much. And thank you everybody for coming. This is just a small part of what we've been doing at the University of Minnesota dealing with PFAS. As you might imagine, with 3M in our backyard, we sort of have quite a few interests in these chemicals. So, but what I'm going to talk to you about today is just some work that we've been doing looking at different remediation technologies and sort of the evolution of how we came up with this, because it's really all based on environmental chemistry. This, as I mentioned, all started several years ago. This is back when ES&T used to be hardcover or hard copy, and you can actually get it. There was an article in it talking about the growing concern over perfluorinated chemicals. Back then we called them PFCs. Now the term we use is PFAS, per and polyfluoroalkyl substances. And one of the biggest things, of course, was 3M Scotchgard. But these chemicals were used in a variety of materials, paints, coatings in textiles, your food wrappers, microwave popcorn bags. Since then, a lot of these chemicals have been removed. Some have been replaced by other chemicals. If you learn nothing else from my talk today, it's don't eat microwave popcorn. So we started looking at these chemicals in the environment, and as I mentioned, I'm an environmental chemist, and so what we do is figure out processes, not necessarily studying the chemicals, but looking at how they move through the environment. And one of the things we want to look at is if you had these chemicals in a, let's say, an aquifer or groundwater situation, how would they partition to mineral surfaces? So this was a paper that we published back in 2007, where on the y-axis we have the amount of PFOS absorbed per surface area versus equilibrium concentration. These are just absorption isotherms. And we did it for goethite, high iron sand, Ottawa sand, and kaolinite clay. Interestingly enough, you get different types of curves. Low enough, you get you're in sort of the linear isotherm region. As you get higher concentrations, you can get things that approach Freundlich or Langmuir isotherms. So we started looking at this, and this was just a simple system of dissolved PFOS and mineral surface. So it doesn't really necessarily mimic what we see in the environment. When you look at partition coefficients in the environment, they could vary over an order of magnitude or 2 orders of magnitude. But interestingly enough, when you normalize to organic carbon, the log KOCs collapsed around about the same number, around 2.8, anywhere from 2.4 to 3.1. These are chemicals that were used to repel oil and water on your pants, your carpet, and your McDonald's food wrappers. Why on earth would they partition to organic carbon? Well, you have to think about how they are the difference between being attached to a surface and being free floating. I'll show that in a second. I had another student who was looking at partitioning in the environment and saw that we were getting much more PFAS in the particle phase than we would expect. This was in a stormwater system. And he started saying, well, you know what? When you guys published that paper in 2007, you were just looking at pure water. What if ionic strength plays a role? Of course it does. And so he looked at the partition coefficient as a function of salt concentration, sodium chloride. And he looked at it in just a single PFAS compound study as well as a multi-compound, putting in more than one PFAS in there. And you can see you have competition. And the effect of the salt concentration goes down as the competition is included. You don't have as much of an interesting situation. And so, of course, we always think, all right, when you hear hoof prints, think horses, not zebras. So they said, well, maybe what we're doing is just by introducing the salt is we're just salting out the compound, basically reducing the solubility. And so for PFOA, this is actually what we would predict for the change in KD with the salting out effect, but this is actually what we see. So he started thinking about this and trying to figure out what the heck is going on. He actually then calculated the free energies of adsorption for several compounds, from perfluoroheptanoic acid up to C11, undecanoic acid, and again saw an increase, or excuse me, a decrease in the free energy of adsorption with increasing salt concentration, increasing sodium concentration. And he had lots of different theories for this, which we can get to in a second. Back to thinking about these molecules, why are they behaving this way? Why are they going into organic carbon? Why are ions and things affecting their partitioning? If you look at the PFOS molecule versus just the straight alkane sulfonate, so all hydrogens, the hydrocarbon, versus the perfluorochemical, what you see here is the blue color indicates negative charge, the red indicates a positive charge. And this is based on a molecular model that we had run. And what you see is the fluorines are so electronegative that it pulls the negative charge away from the carbon backbone, whereas in this molecule, just a hydrocarbon, the carbons are slightly negative and the protons are, not surprisingly, the hydrogens are slightly positive. It flips. So what that means is the entire surface of this molecule is negatively charged. Much different than most surface active molecules that we think about that have a really hydrophilic head and a hydrophobic tail. Okay? So now instead of looking like, and if you were to attach this then to a surface, what you have flailing up into the aqueous system is a fluorinated hydrocarbon, or not hydrocarbon, fluorocarbon, which is going to repel the water and the oil. But when you break it off, the pKa of this, by the way, is less than 0. Nothing really wants to stick to the end of that. This is an ion. This is why you don't find them in the atmosphere, despite some people saying they're finding them in the atmosphere. They're not finding them in the gas phase, that's for sure. This can then freely dissolve. So this is a molecule that has a molecular weight of 499, but has milligrams per liter solubility. That's a little sketchy. Trying to figure out a solubility of a surface active molecule is a little difficult because you can get micelles, how many micelles forming. You can actually form a third phase. You can't actually even calculate or measure. You can't measure a KOW of this molecule, because what it will do is, if you know how to measure a KOW, you take octanol and you take water in a beaker and you put your chemical in there and you see where it partitions. You do that with this, it forms a third phase. So KOW, octanol water partition coefficient, going into lipids of biota is completely a non-starter for this chemical, which is what makes it so interesting as an environmental chemist. Because as I said before, we were studying processes. Well, these molecules behave so differently, such different processes, that we have to think about them differently. So my student was saying, all right, these things are basically like little negatively charged particles out in the water. They aren't particles, they're very small, they're molecules. But what if we treated them like particles? And I'm jumping ahead. And one other thing I wanted to say before I got to that, sorry. One of the things that we can do by attaching, so one of the thoughts was with the salt, are we affecting not the molecule, but the surface that we're absorbing to? And so we did some mobility zeta potential calculations, so basically surface charge calculations. And what you see is when you attach PFOS to kaolinite, you decrease that surface charge. So you have a negatively charged molecule, you attach it to the surface becomes more negative. Not really super surprising. But it's also, again, effect of salt concentration. The higher the salt concentration, the higher the negative charge, because the more PFOS is going on there. And you can look at, this is kaolinite surface charge. If you put PFOA on, perfluorononoic acid, decanoic, undecanoic, you can see as the carbon chain gets longer, we get even stronger negative charges. But PFOS bumps back up, which is a sulfonate. Interestingly, PFOS binds to the kaolinite much stronger than these other ones, but doesn't affect the surface charge as much. So again, sorry, backing up to what I was trying to say before, my students started thinking, okay, what's going on with this material? You add the sodium, basically you could have bridging, right? So you got negative charge and negative charge, you got the sodium in between, it's bridging it. You have all kinds of other things that are going on. He said, what if you added a coagulant? And so he did this in a water treatment type system, adding alum. And as you, and this is the percent removal of PFAS, PFOS and PFOA, PFOS being the blue triangles, PFOA being the, is it black or red? Thank you. Red circles. You can see that as you increase alum dosage, you increase the removal of PFOS and PFOA. More interestingly, the turbidity goes away very quickly. We're removing the particles with the alum. Alum is a coagulant. Coagulants overcome electrostatic repulsion among like charged particles. We're getting rid of the particles right away. So what's going on here with this increased removal of PFOS and PFOA is not just getting them to stick to those particles and the particles falling out. Something else is going on here, okay? And so he did a bunch of more work and tried to figure out what's the optimal conditions. So here we have a PCPH diagram, and within it you have percentages of removal and also alum dosage here on the other y-axis. Excuse me. And what you can see is if you get low pH, really high alum dosage, you can get up to about a third of that PFOS and PFOA removed. So we thought, well, that's kind of interesting. Now, 30% removal is not great. We got better than that removal if you're just looking at a groundwater system. You got about 50-50 distribution. So if you just added dirt or sand, you could get about half of it gone, right? There's just not a lot of particles in this system to help us out. But then we thought, well, what if we combine the two systems? And this all came about because there was a call from the Department of Defense to look at in situ remediation of groundwater contamination with PFAS. The reason the Defense Department is interested in it is because these chemicals were used in aqueous film forming foam. Aqueous film forming foam is a material. It's since the 60s, it's fluorochemical based. It forms a foam, just like its name implies. You spray it out onto a hydrocarbon fire. It does two things. One, it starves the fire of oxygen. The other thing it does is it cools the fire very rapidly. The heat capacity of things is very high, and so it cools the fire faster than just about any other material they use. Before they had fluorochemical-based AFFF, they used AFFF that was based on fish scales. So again, a surface active molecule made of foam, but it didn't cool the fire as fast. It didn't quench the fire as fast. Didn't put out the fire as fast. If you're a military installation and you've got a fire on, say, an aircraft carrier of jet fuel, you got to get that thing put out right away. So this stuff worked really well. The other thing that the military likes to do, besides saving its people in an emergency situation, is they like to practice. So that could be as complex as a concrete pad with the skeletal structure of an airplane that they light on fire with jet fuel and put out with firefighting foam, to, hey, there's a field, let's dump a bunch of jet fuel, light it on fire, and put it out. So the Air Force alone has 374 sites around the country that are contaminated in the groundwater with PFAS from AFFF. They are they have committed to following CERCLA laws, which is good, super fun laws. So they have to figure out how to clean up all these sites. And so they said, look, we want a way to clean this up in place. And so we came along, or they wanted a way to destroy these molecules underground in place. And we came along and said, look, you can't destroy these things in the lab. There's no way in hell you're going to do it underground. But we might be able to combine the two studies we just did and see if we can't get it to be more sticky and get it to bind to the solid material and allow the groundwater plume to flow through uncontaminated. And so we said, all right, we played around with these coagulants. They seem to work. We know that if it's in a groundwater system, it likes the particles if a little bit. If we combine the two, maybe we can get it to work. So we started thinking about, okay, what kind of polymers, or what kind of coagulants do we want to use? And we basically chose 4. One was polyaluminum chloride, which is just a regular, kind of like the alum or ferric chloride that you would see. It's basically just increasing the ionic strength, basically. Another one was a tannin-based coagulant. Tannin and tanninic acid is like natural organic matter. These things like organic carbons. We thought, oh man, that would be great. That was a disaster. It just gummed up our instrument. And after about a month, I said, put that on the shelf. Don't ever touch it again, because it was such a nightmare. But we had 2 of the other 4 that actually worked pretty well. All of these, by the way, were cationic polymers, or cationic coagulants. That's because the PFAS are all negatively charged. All of the ones that we're looking at. There are certainly zwitterionic and cationic polyfluoroalkyl substances as well. But we didn't even know about those when we wrote this proposal several years ago. And the 2 coagulants we found that work really well. One is polydadmac, which is I can't remember what the DADMAC stands for, but basically you've got a 5-member ring with a quaternary amine group on it, and this is on the order of 2 to 3 million Daltons, a big polymer. Okay. The other one is polyamine, and so in this case you have, again, a quaternary amine here and an alkyl group, and this is about 750,000 Daltons. We wanted to do different size groups to see what might work. So the first thing we did was we did a bunch of batch studies and looked at adsorption isotherms, just like the ones you saw earlier. And so what we have here, again, on the y-axis is the amount absorbed to the soil, and the equilibrium dissolved phase concentration on the x-axis. The top 3 panels, we were only looking at the 6 compounds in the UCMR3 list. The UCMR3 list is the unregulated contaminant monitoring rule 3. Now, if you do a CERTIT project or anything, you have about 19 compounds you need to look for, because the EPA came out with a new list that they want everybody to look at. All right, so what you see in all 6 panels, so the top 3 are the sulfonates, the bottom 3 are the carboxylic acids. The dark circles you see, the solid circles, are the controls. So this is just absorption to Ottawa sand without any modification. The other 2 are either polyamine or polydadmac. And you'll see that we drastically increase the absorption of PFOS, PFOA, perfluorohexane sulfonate, perfluorobutane sulfonate, heptanoic acid, and nonanoic acid. So we also got some soil from an actual Air Force base that was contaminated, Tinker Air Force Base. Notice here that we go anywhere from 0 milligrams per gram of soil up to 35 milligrams per gram. We go to Tinker Air Force Base, and our y-axis jumps up an order of magnitude. Now we're looking at 0 to 300 for PFOS. But again, the solid is your control, and the other ones are your polydadmac and polyamine, drastically increasing the absorptivity. So we said, okay, that's a batch system. Let's look at a 1D column system to see how this might actually work in a groundwater type system. So we do a 1D column, and what you see, again, dark circles is your control. We get breakthrough right away of perfluorobutane sulfonate. That stuff is horrible. Hexane sulfonate a little bit longer. PFOS still on the order of about 5 pore volumes, and it's popping through. We add polydadmac or polyamine, and you can see that we go from, in this case, 5 pore volumes out to 20 pore volumes before we start to see breakthrough. And we start we delay the acids as well. But interestingly enough, this PFOA is not increasing like we expected it to based on, based on the batch experiments. So something was going on here. We're not getting the retention we wanted to here. So we're trying to figure out what happened. I forgot to preface, the way we built these columns, we had the columns, we loaded them up with our coagulant, and then flushed through contaminant-laden simulated groundwater. Okay, so we loaded up the soil first with the coagulant, then we passed our PFAS through. So we were wondering, is there something else going on here? So is this a kinetic thing? We're just pushing the water through too fast, we're not seeing it come to equilibrium? Or is it the fact that the coagulant is stuck to the soil that's causing a problem? And in the batch experiment, of course, everything's all together. Could we be getting some sort of complexation of the PFAS with the coagulant, and then that's what's sticking to the soil? So to test that, we took a column and we had an extra couple of injection ports put in. And what we do is we flow PFAS through, in this case PFOS on the top. We start flowing it in, and at some point we start injecting, and we see breakthrough. Same thing with PFOA, because Ottawa sand is not very retentive. And then at the dotted line, we start injecting through a side port the polydadmac. And so what's happening is our PFOS is going away, our PFOA is going away. It starts to break through a little bit later. But we're pretty sure from this, it's leading us to think that we're forming some sort of complex with the PFAS and the polydadmac. And then that's what's sticking to the soil. So to test this, it's very hard to do, because if you take your solution if it's forming a complex and then you say put it on your instrument, as soon as you get into that organic solvent, you're breaking up that complex and you can't see it. So one way we can do that is with fluorine NMR. And oh, this is a little fuzzy, I apologize. This on the top is perfluoroheptanoic acid. We have the carbon numbers 1 through 6, and here are the fluorine peaks associated with them. If we add polydadmac or polyamine, notice first that the peaks get smaller. This is semi-quantitative, and they also shift. Okay? If we look at that shift from the carbon number from 1 to 6, you'll see that as we get further away, or excuse me, as we get closer to the sulfonate group, we get a greater shift. Okay? What that's telling us is, I take it back. I said it right the first time. As we get further away, as we get further away from the functional group, in this case PFOA, the carboxylic acid, we get greater and greater shift, which is what that's saying is then what's sticking into our coagulant is the tail end, and what's sticking out is the functional group. Not too crazy if you think about a surface active molecule. You'd expect the sort of hydrophobic tail to be in with the organic phase. Okay. Well, what's more interesting, this is heptanoic acid. If we look at PFOA, all the fluorine peaks disappear, both in polyamine and polydadmac. What that means is we're getting such a great shift that we're not seeing it, or it's binding up so strongly with that coagulant that it's not even resonating in the NMR, meaning it's not coming in and out of solution, in and out of that complex. It's binding really strongly with that complex. Okay. So if it's binding really strongly with that complex, perhaps we'll have some hysteresis in desorption. right? So we adsorb onto the solid phase. Could we have some hysteresis? So what we did is we took the columns, the Tinker soil columns that we had loaded up with coagulant and PFAS, and then started flushing it with clean water just to watch it come back off. We do that here. This is the control, no polydadmac. You can see we got breakthrough right away again. We've definitely saturated the column with PFAS. We start flushing with clean water, and we get everything back off. Polyamine, not quite so good. Polydadmac comes right back down. We can actually look at the area under the curve and figure out what we've actually gotten back off the column. And for our controls, we're getting 100% back off. From polydadmac and polyamine, certainly for PFAS, we're getting less than 10% back. So what we have is something that will bind very strongly with the PFAS, stick to the soil, and not come back off. Okay, the next question is then how long is that going to last? People asked us, what about these polymers? Can they break down? Of course they can. So what we did was we looked at, well, first we inoculated Tinker soil with our coagulant, so an actual soil from the environment. Didn't get any breakdown whatsoever. So we thought, well, let's hit it with the best stuff we know of. Let's hit it with activated sludge. So we inoculated activated sludge with the coagulant and the coagulant PFAS mixture. And what you see up here is just the control, no biological activity. Then what you see here in the triangle is polydadmac and activated sludge. So it does break down the polydadmac. Could be good, could be bad, we're not really sure. Interestingly, when you combine the polydadmac with the PFAS, it also degrades polydadmac, but not as well as it does the other one. And we do the math on this. It turns out that what's not being degraded is the fraction that's bound up with the PFAS. So the coagulant's biodegradable unless it's bound up with our PFAS. So just to reiterate, we've got something that will bind very strongly, won't come back off, and won't degrade within the time frame that we've looked at. Everybody always asks, how long is it going to last? We have no idea. But we want to find out. Keep funding me for 20 years and I'll tell you. Right? Okay. Now, it turns out that going back and looking at some of these other, the Ottawa sand, we're still getting some breakthrough. Ottawa sand, if we get into a situation that's a really sandy soil, we're going to improve things, but it may not be as good as we want it to be. So we had my colleagues at Tufts University decided, let's play around with some other stuff. We've got some free time. So they said, what about activated carbon? So they took and did something really, really interesting. They weren't just thinking about adding powder activated carbon. They were thinking about combining it with this coagulant. So in this figure here, you've got just activated carbon and water, and they sonicate it. So they mix it all up so that it's a suspension. Do the same thing with polydadmac and powder activated carbon. Looks the same. And after 48 hours, notice the stuff without the coagulant settles out. The powder activated carbon stays in suspension. In fact, you can take that and centrifuge it and it doesn't come out. What we've done is we've created an injectable powder activated carbon that we can put underground. Okay? Our thought process was we could boost what we're seeing with our coagulant adsorption. So here are our columns of Ottawa sand. There's the clean Ottawa sand. We start pumping through the powder activated carbon. We keep pumping it through till we see it come out the other end, and you basically end up with powder activated carbon and sand. Now, we had to make sure that what we weren't doing is just plugging the hell up, plugging this up with powder activated carbon. And so we did some scanning electron micrographs, and what you'll see is we're actually not just throwing in powder activated carbon. The coagulant is allowing that powder activated carbon to adsorb to the surface of these sand particles. So we're taking a really low organic carbon sand and creating a high organic carbon sand in place. Okay? And if we do this and look at the breakthrough again of PFAS, you'll see here we're pumping in 100 micrograms per liter PFAS, which is a pretty high groundwater concentration. And for 20 pore volumes, we don't see any breakthrough. So then they said, oh crap, we're not seeing anything. Let's bump it up to 100 milligrams per liter. And in another 5 pore volumes, we finally started seeing stuff come out. We can do the batch experiments to figure out the capacity of these columns. And it turns out it's about 4,300 pore volumes at 100 micrograms per liter before we see breakthrough of a sandy soil that would have no retention of PFAS prior to us putting anything in. This is all in the lab. We still want to try this in the field, and we've applied for funding to do so. Okay? So, just to reiterate, we have something that binds strongly to our PFAS. It sticks irreversibly to our soil. It doesn't break down, and we think we can make it even better by adding, one caveat to this. If we just looked at the capacity of powder activated carbon, the amount of carbon that's on there, powder activated carbon is doing all the work here in this situation. So in our previous studies where the coagulant complex was aiding us, in this case it's basically the activated carbon. But we're only flowing in PFOS to a soil that's already pre-treated with the organic carbon, activated carbon, excuse me. What we'd like to do is, the next step, and we'd like to do this in the field, is upstream of this, inject our coagulant to see if we can form that complex, see if that complex has a greater affinity to activated carbon than the PFOS itself. So sort of combine the two discoveries together. Okay. So the first conclusion we have is that PFAS are weird. If you didn't get that, I can talk to you at hours of why these molecules are weird. They don't behave like PCBs, and all of what we built our body of knowledge in environmental chemistry on. These guys don't apply. They don't follow the rules. Well, they follow the rules. We just have to we have to understand the rules better. But the beauty is we might be able to use that weirdness to our advantage. The fact that they have that entire surface of their molecule negatively charged, we can take advantage of that to use it to make it stickier. So we've successfully developed a PFAS sequestration in a groundwater system. The next step is we'd like to try it in the field. So we're right now being considered for funding by the Defense Department to try this out at Wurtsmith Air Force Base up in the Oscoda, Michigan, northern Michigan. That system is a very sandy, low organic carbon soil. So we'd like to try the upstream coagulant, downstream permeable absorptive barrier of polydadmac and powder activated carbon, and see if we can actually prevent this plume from intersecting an adjacent reservoir where, when the wind kicks up, foam reforms on the lake. And they've, of course, found PFAS in that foam. Now, I've argued that I don't think it's actually firefighting foam that's forming on the lake. It's probably natural organic matter that's foaming up, but it just happens to be that there's PFAS in there. Explain that to the people who have cottages on that lake. The other thing is, could we use this coagulant and powder activated carbon in other systems? So other systems being drinking water treatment. So I showed you the increased retention of perfluorobutane sulfonate. That is notorious for breaking through activated carbon. The city of Oakdale gets about 6 months of their granular activated carbons before they have to regenerate and replace their activated carbon. Those things should last 3 to 5 years. So we're wondering if combining our coagulant with the PFAS upstream will increase the bed life of that granular activated carbon. Wastewater treatment plants. These things were used everywhere, right? So they end up in our waste stream. You could buy that can of Scotchgard for a reason, right? When you bought furniture, you could have it Scotchgarded. That was like an option, right? Well, you could also buy a can of Scotchgard to reapply it later. Why did you have to reapply it? Because it wasn't there anymore. Well, that ends up in your waste stream. So it ends up in our wastewater treatment plants. That could be a concentrated stream where we can deal with it as opposed to then spreading it back out. You know, when I first got into environmental chemistry, we'd already closed off all the pipes. You know, Lake Erie was no longer dead. You know, it's easy to go after a sewage pipe going off into a lake. It was all about non-point source pollution. Well, why not then go to these places where we have these chemicals going through a narrow point, like a funnel point, as opposed to trying to deal with them out in the environment? Let's deal with them where they are more highly concentrated. So we'd like to try it in a wastewater treatment system. That's got a high solids, suspended solids in the activated sludge area. Can we, number one, can we reduce the PFAS in that stream? Number two, can we do it without screwing up what the wastewater treatment plant is trying to do, which is remove biochemical oxygen demand and solids and phosphorus and some other things. But they're not designed to get rid of these micropollutants like PFAS or microplastics, which can carry a negative charge, or pharmaceuticals and personal care products, which can carry a negative charge. Or can we use an anionic polymer coagulant to get rid of some cationic compounds? So those are things we'd like to try and do. Or landfill leachate. Oh, I mentioned other chemicals or substances, so microplastics or pharmaceuticals, personal care products. Or landfill leachate. We've got a proposal in to look at applying this in landfills. Again, you've got it in a concentrated area. What most landfills do is they collect, easy for me to say, collect that leachate, put it in a pond, either let it evaporate away, or they then spray that on a field. Let's not spray that PFAS-laden leachate out into a field. Let's deal with it while we've got it in a system we can deal with in a pond. Okay. I mentioned that this doesn't break down, and it's very strong, and it seems irreversible. What if by adding these coagulants we can complex up these PFAS, remove them in the activated sludge, that activated sludge becomes biosolids? What if applying that biosolids renders these compounds not bioavailable for uptake by plants? Then we can still land apply biosolids. We don't land apply biosolids in the metro, in Twin Cities metro area. They incinerate their biosolids. Almost all of them. There are some plants that create biosolids, and they do land apply, despite them telling me that they don't. But they do that incineration for several reasons. One, it's for energy cogeneration. So it's basically as a feedstock to their boilers to heat their plant. And it's also then a lot cheaper than trying to truck it off and deal with it, especially if it's got high contamination in it. So I apologize, that was kind of a breeze through really fast of what we've done and how we sort of came up with it. Now we're just trying to figure out how good is it and where can we apply it, and hopefully actually try it in the field. I will mention, we were talking about this earlier, what we're trying not to do here is create a new problem where an old problem existed. We realize we're pumping something underground. We want to make sure that that's not an issue. The coagulants we chose were commercially available drinking water treatment coagulants that are on the market, because we figured if they're using them for drinking water treatment, either they're safe or we know how to deal with the challenges. So polydadmac does have an issue if you then chlorinate the water to disinfect it. You can create NDMA, nitrosodimethylamine, disinfection byproducts. But we understand that. So we know that that's a challenge and we can deal with it. Yeah, we're hoping that we're very cognizant of the fact that we don't know how long it's going to last, and we want to be very aware that we're not creating a problem. We're replacing one problem with another. Okay, thank you very much.
Thank you, excellent talk. We'll take questions.
Oh, I forgot to acknowledge all of of course, I didn't do any of this except choose the coagulants. I got lucky. My PhD student, Yosef Ali, did most of this work, postdoc Dan McInnes, Bill Arnold in the civil engineering department, and then Kurt Pinnell's group at Tufts, who's now at Brown, his PhD student, Chen Liu, did all of the powder activated carbon along with Bonnie Lyon, his postdoc, sorry, and funding from CERTIP. Sorry about that.
Okay, well, thanks for the talk.
Mm-hmm.
Very, very interesting. I wondered when you mentioned about spraying the leachate, are there rules that you know of in Minnesota or elsewhere about testing the leachate before they it's better, because one of our concerns is with all of this possible irrigation of fields with water that might have pharmaceuticals in. And so we were just wondering, do they require testing or maybe not of emerging contaminants?
Yeah, they are in Minnesota, and I know they're doing it for PFAS because we have one landfill in St. Louis County that is no longer going to be allowed to spray their leachate. They're going to have to ship it or truck it or pipe it down to the wastewater treatment plant in Duluth, which is a long way away and cost prohibitive. So they are very interested in us coming up to try and see if we can remove it in their ponds so that they can still spray. And yeah, and of course, wastewater treatment plants are not going to want to take a lot of this stuff at some point. And so the poor landfills are going to be sort of between a rock and a hard place. They can't apply, they can't send to a wastewater treatment plant. They're going to have to do something with it. And it may be as simple as just adding activated carbon to your pond. I don't know. That's 3M. One of my best friends is the director of the environmental group at 3M, and he figured out, he actually used polydadmac in one of his tests in their wastewater treatment plant. He goes, yeah, it'll work. But they just decided to use powder activated carbon because it was just cheaper for them to do. When you're dealing with a waste stream and you only have a few components in it, activated carbon will work really well. The problem with landfill leachate is you have such a diversity of compounds that activated carbon might get overwhelmed. And so we're hoping that the coagulant will sort of help us out there and take advantage of all that other stuff. And so our proposal is actually to deal with it in a pond, deal with it, inject it into the landfill itself to see if it can't stick to the garbage that's in the landfill and not come out. Mix it in with the daily soil cover maybe, so that when it leaches down through, it sticks to that one of those, and figure out how many layers we'd have to do to basically trap that in there, and so that we're not, so that these are the final resting place for these compounds and not come back out. That was a long-winded answer to a short question, sorry.
I'll remind our online audience that you can type in your questions at any time during the question session, and we'll get to yours in a couple minutes. And we have
actually, I'm just curious how you can handle the collected or the filter, the PFAS. I mean, after collecting all the PFAS and you're
right. So
burn them out, or just reuse it, or I'm just curious.
Right, and that's the question of then how long does this last type of thing. So what we're basically trying to do in the groundwater system is we're trying to contain that plume. And for the Defense Department, if they can contain the plume and keep it from going off base, then that removes their liability, right? I think they're just happy to leave it where it is. The alternative is what they're doing now, is excavating and having to deal with all of that, which is extremely expensive. In the end, if we're creating let's say a 10-meter permeable absorptive barrier, we've at least reduced the amount of material that would have to be excavated and most likely incinerated. That's our hope, our goal.
So do you know what happens to the PFAS in incineration? And these are highly thermally stable molecules. Have we really solved the problem by incinerating?
That's an excellent question. So I know that the wastewater treatment plants in Minnesota, I think, are on the order of 600 C. Is that right? That doesn't sound right. I shouldn't give you a number because I can't remember. Whatever number it is, they have looked at the literature and said that is going to destroy it. I don't know exactly how they came up with that. We offered to see if it was working. They said no, thank you, because they're just operating on the assumption that it's working, and they don't want to find out otherwise because they're not being regulated for it. The other issue is a regeneration of activated carbon. So at what temperature do you reactivate that carbon? Are you just desorbing the PFAS? Are you even desorbing the PFAS, or are you destroying it? Luckily, in drinking water treatment, you can only use virgin GAC. You can't use regenerated GAC. So it won't be an issue for recontaminated drinking water. But if there are landfills. So the alternative for that St. Louis County landfill is to put in a wetland to take up a lot of the material and then go through GAC before they land apply or land apply. That could be regenerated, and you could use, presumably you could use regenerated GAC in that. And the question of whether or not that actually works is an open question. We started looking down it until we were told, no, you can't use any regenerated GAC in drinking water. Because then we thought, well, you know, how would our coagulant complex absorb to the GAC affect that regeneration?
A comment on that, you can use regenerated GAC if you do it on site. So Cincinnati has their own regeneration facility and regenerates.
Oh, if you regenerate the GAC on site.
If you don't, you can't take it off site back to Calgon and then bring it back, but you can regenerate it because then you have your own contaminants. The idea is you don't want to.
I see. So you're not sharing your contamination with other people.
Right.
Oh, but can you regenerate it as well as Calgon can?
Yes.
Okay.
Yeah, they have the same equipment. That
the same equipment.
uses.
Okay.
You just
I was under the assumption that everybody just sent it back to Calgon.
Facility. Yeah, you just need a large facility.
Okay. Well, then that would be an interesting thing to look at to see if that's even working. Thank you for that.
Yeah, good talk. You know, I wondering if, because you show me a lot of your lab experiment, you know, bench sorption and the column sorption, you know, column leaching experiment. I wonder, I assume you do the experiment, you're using high concentration in the PFAS to work on this for the, you know, bench sorption, our leaching.
No, we used environmentally relevant concentration. So the one
nanogram per liter level.
Yeah.
Okay. So this is all the low concentration level.
Yes.
To remove.
Right.
Okay.
Right. That's one of the reasons CERTIP likes us, is because we're doing things at environmentally relevant concentrations. Yeah.
Okay.
Yeah. Some of the so the one that was really high was this one, is because we were trying to overwhelm the system. But these other ones, we are, where are we? All the way back here. Well, we're micrograms per liter, but we're down. Whoops, wrong button. We're down around, you know, 5-10 micrograms per liter. So we're a little bit higher.
Yeah, because I pasted this one all the PPB level, you know,
Yeah.
micro. I saw the nanogram should, you know, PPT level. I saw.
Right. So yeah. So we're not talking about trying to treat surface water. We're still talking about contaminated groundwater here. These are relevant to contaminated groundwater systems, not your surface water systems.
What's the concentration rating for the contaminant, you know, for the groundwater, contaminated groundwater?
Oh gosh, it can go up to thousands of micrograms per liter.
Thousands.
Yeah. So parts per million level. Part per million
in the
level.
water, in the groundwater.
In the groundwater,
in the groundwater, okay.
Where you have AFFF real close to the source, you can get very, very high concentrations. Yeah.
Okay, got it.
But this is more in the range here of 10 to 100 ppb is usually what you'll see, and that's why we were at that 100 microgram per liter, because that was sort of a typical contamination site. Whoops. That's why we chose that 100 microgram per liter, because that was relevant to some contaminated groundwater systems. And what we're seeing, what has been monitored up at Wordsmith Air Force Base, where we like to try this, it's upwards of that, and even an order of magnitude higher than that. They're up to milligrams per liter.
Okay, thank you.
Yep.
Other questions?
One of your breakthrough graphs that was slightly before one of the fuzzy blue ones, yeah, probably here. It looks like some of them come down before the DADMAC or the other polymer was added. Do you have any explanation for why that would occur?
It could be just an error in where I put the dotted line, but that's a good question. That'd be a question, again. That'd be a question for my student. Well, it's not even my student; it's Kurt's student. I got to find out exactly when they started to make sure that that's right. This is well, no, wait. This one is published. I'll have to look at the paper to see where we said that. But I thought it was on the order of three, 4 volumes. So it would be right in there. It shouldn't be, because it broke through and it's coming through pretty good. So my guess is I just put the dotted line in the wrong spot. but good catch.
Sometimes natural systems have variation.
Yeah.
Really a lab-based system, right?
Right, this is a lab-based system, yeah.
I just follow this, follow this graph. What's concentration for this one?
Oh,
So,
I believe this was 100 micrograms per liter as well. I don't know off the top of my head. It's in the paper, and I can send that to you for sure.
I just wondering when you collect the fraction, you just direct analyzer, or you need to SPE concentrate?
No, we're direct. Yeah, we're direct injecting. Yeah.
For the LC mass.
Yeah.
So this maybe like a PPV then. Okay.
Yeah, good question. Yeah, and yeah.
What's the unit of the pore volume?
So pore volume is the volume of water to flush through one porosity of the column. So if this column is 2 inches by 10 inches, you figure about 30% porosity. So you take 30% of that volume. That's a pore volume. So it's the volume of water that's contained in this, and that's a known number. So we do pore volumes as opposed to volume of water, because it's when you do columns and things like this, it makes more sense.
So are you going to look at the microbiome in the soil? Do you plan on doing
That's
that?
a really good question. Yeah, we actually got some funding to, and the argument for funding us was that this could affect the microbial community. I'm a chemist. I hate biology because it's too complex. So I try and stay away from it. But that is a good question. That is something that maybe I'll mention that to ESTCP, that they should hire a biologist to take a look at it before and after. It would be interesting to look at. Yeah, but of course, in the Tinker soil, we just didn't see much microbial activity at all. And again, these are so the PFAS in this case probably isn't affecting the microbial community because it's such low concentrations. But the coagulant we're throwing in is significant, and so it could. That's a really good question. It's something again, as a chemist, I didn't think about. I should have.
Do we have additional questions from the room? I don't see any questions online. I'll ask a question. At the beginning, you mentioned that PFAS won't be found in the atmosphere in the gas phase, but I'm wondering if particles could have them. Okay.
Yeah, absolutely. Yeah, because so when you're, again, fluorochemicals being weird, if you have a neutral molecule and you put fluorine on it, my inorganic professors love to say that fluorine gives compounds wings. It makes them more volatile. But in this case, when it's ionic, when it's charged, it's just not going to be in the atmosphere. It's going to stick to stuff. So it could stick to particles. Those particles could end up in the atmosphere. Those particles, and if you've got, if it's attached by the functional group and you've got a fluorinated tail sticking out, it might actually even repel stuff. It might make particles even more volatile, like microplastics and things like that. You never know. You see lint floating through the air. Maybe growing up, staring out the window with the light coming through, watching that lint, maybe it was PFAS all in there. I don't know. It was certainly with Scotch guarded material that was coming off of.
Okay, we'll formally close the session. Please
Thank you.
Matt again.